So the other definition we need to work into is herd immunity. All right, so herd immunity, this is the definition that was used by the World Health Organization until November of last year. So before this, we always recognized that herd immunity had something to do both with a vaccine or through previous infection or natural immunity. And for some reason in November of last year, the World Health Organization dropped that part about natural immunity and recovery. The World Health Organization does not speak for the entire scientific community regarding the definition of herd immunity. To ignore that somehow or other previous infection and the immunity you get from that has no contribution to herd immunity is frankly fantastic. It has no basis in reality. So herd immunity should be thought of as a passive description of the average immune competence against a pathogen for the entire population. All right? It's kind of an average of individual cases of herd immunity, all right? And it is a passive thing that you get, all right? It is not an active thing that you must go because you get it individual at a time. The other thing you need to know about herd immunity, it is not defined relative to the endemicity, pandemicity, or epidemicity of the pathogen we're talking about. So it's not like, geez, if you can just get this point in herd immunity, the epidemic is gone. That is not any equation that have these two concepts that are put together. And there's been a lot of browbeating out there about, oh, if we can just get to herd immunity, we won't have a pandemic or an epidemic anymore. They are unrelated concepts. The other thing you need to know is it's a graded phenomenon. It's not black and white. It's not like, geez, if we can just get one more person to survive an infection or one more person to get a vaccine, we'll have the magic thing called herd immunity. Herd immunity comes in degrees. The herd immunity to influenza is pretty good in the summer, right? In the winter, herd immunity is not so good. And that's the last thing you need to know about herd immunity is it's not static, right? It doesn't stay the same all the time. So you can have a good amount of herd immunity. In fact, one of the things that made smallpox go away was improvement in nutrition that made it so that the immune system was better at fighting the virus. So there's a bunch of ways that you can contribute to herd immunity. One of them is you can die from the disease. And this isn't the favored way to most people, but we need to acknowledge that if you get the disease, if a human being chooses to say, I'm just going to do nothing and get this disease and you die from it, you still contributed to herd immunity. So there's been a lot of guilt therapy out there about, hey, if you really care about the population, you're going to go do this. Well, that's just not a real thing. If you just choose to do nothing and die, you still contribute to herd immunity because there's one less person in the population who can spread it. The other way you can do it is you can recover from natural disease, and you're going to hear a lecture right after this about all the different things we have to protect people so they can survive this disease when they do it. A little bit better is if you can actually get the immune system so good, you can actually get natural immunity with an asymptomatic infection. As a matter of fact, that's an important thing people should realize about most of these respiratory viral pathogens, and that is that about 50 to 70% of the people who get influenza, common cold, COVID-19 have no symptoms at all. Now, that's important because when we start to do the calculation of disease, we end up thinking like disease equals the pathogenicity of the bug divided by the quality of the immune system. And I can tell you we have a tendency to focus on that thing of, gee, how bad is the bug, without ever asking the relative question of how good is the immune system. And then the last way you can get herd immunity is that you can get a vaccination. Now, vaccination, I am not an anti-vax people. I've said it before. I'm glad my mom and dad gave me MMR. I'd give it to my own kid. But people didn't know there are some realities of vaccination. It's good at preventing disease, but it's not as good as natural immunity, and that's true for every vaccine I've ever seen. How do I justify that statement? If your kid gets measles at the age of 2, his chance of having measles again in his life is extremely small. It's less than 1%. But if I give you MMR vaccine at age 1, your risk of having disease is low until about age 5, and then it starts to go up because you lose immunity. It doesn't last as long. And I have to give you 2 exposures to the bug, another booster at age 5, to get you the same level of immunity that you had from natural infection. So it should be understood from the get-go that when we're manipulating the immune system with a vaccine, that you don't get the same quality of immunity in any disease we've ever looked at as if we have you recover from the infection. So while there are people who are saying that those first 3 options just don't contribute to herd immunity, that's not true. The other thing you need to know about herd immunity is it's not something that may or may not develop. There is always some degree of herd immunity that develops, and the best proof for that is you're not all dead yet. All right? If these viruses were all that in a bag of chips, the human species would have been wiped out. But we have immune systems. So, with that said, I'm going to do what I do best, which is bore people for a living, because there are some basics of immunology that I think are going to make it so you can participate in this discussion once you know the rules of that. So, these are some cells that line your respiratory system. And the first thing that happens is some virus comes up and you get exposed to it. Now, this is a virus right here, and I don't know if Mr. Pointer is going to work, but those red and blue things are proteins that are made by the virus that are exposed on the outside of the virus. And next step is that virus gets inside your cell. Now, one of the things you need to know about viruses, they can't reproduce themselves. They have to hijack that cellular machinery to do that. But as they hijack the cellular machinery and start producing their own proteins, they could put on the side of the cell, and that stresses the metabolism of that cell in a particular way that we call endoplasmic reticulum stress. And when it gets into plasmic reticulum stress, it makes this little signal that it puts on the outside of the cell to tell all the neighboring cells that, hey, my metabolism has changed in a way that is likely being caused by a virus infection. And the other thing it does is to start to secrete some things called cytokines. Now, cytokines are chemical signals that tell the brain, the immune system, and all the surrounding tissue about the nature of the injury for that cell. All right? You have different cytokines if you get a bruise. You have different cytokines if you get a burn. You have different cytokines if you get different types of infections. And cytokines are also specific to the tissue they're in. All right, so your lungs make different cytokines than your pancreas does. And these cytokines, they do several things. One of the things they do is they go to the surrounding cells and they say, hey, look, we may have a virus around here, so you should change to cell danger response. And in cell danger response, these other cells will actually make their cell membrane become very stiff. They change their metabolism, so they actually become resistant to viral infection. This is one of the key points you should take away about immune system function is this cytokine release. Because when a cell goes into cell danger response, those metabolic changes it makes it do its job much less well. You do not die from COVID-19 or influenza pneumonia because all of your respiratory cells fill up with virus, rupture open, and die. You die because of an unregulated excessive production of cytokines that makes all of your cells go into cell danger response and quit doing their work. So this isn't like a streptococcus infection where you can just have the cells liquefied and destroyed by the bacteria and you'll get sick from that. You die from cytokine storm. So dysregulation of this process is is very important in the generation of disease. The next thing cytokines do is they call in the white blood cells of the particular type that you need to fight the type of infection that you have. So you don't need every type of white blood cell to fight a viral infection. The first guy you need is a natural killer lymphocyte, and the natural killer lymphocyte has no idea what germ he's fighting. That ain't his job. He just comes in and he asks every cell, hey, are you a healthy cell? And when he sees that signal on the outside and says, oh, you're not a healthy cell, he gives some chemical signals to the cell to make the cell commit apoptosis. And apoptosis is programmed cell death. It's a series of chemical reactions that begins to digest all of the genetic material inside of that cell, both the human and the viral. And when apoptosis is triggered, the next thing the cell does is put a little signal on the outside of the cell that says, hey, I just committed apoptosis. And the reason that signal is necessary because the next guy into the party is a white blood cell called a macrophage, and the macrophage is the body's garbage man and recycler. And he gets his signals on what to do from the cell he's taking care of. So Mr. Macrophage goes up and says, oh, I see what's going on here. We happen to have a cell that's got some kind of viral problem in it, and he eats up and gobbles up the cell, and he takes those external proteins that have all the viral proteins and the signals that say I'm in the respiratory system and I've got a cell that's endoplasmic reticulum response and has just committed apoptosis, and he takes that to our friend called the T helper lymphocyte. Now you see that little arc there? That's a specific receptor because T helper lymphocytes recognize one and only one part of one particular protein. They are very guided missiles. But they have to have instructions on what to do when they're exposed to their guided missile. And Mr. Macrophage, by exposing him to all the information that he's gotten from that injured sick cell, makes that T helper cell become a T helper 1 lymphocyte. And T helper 1 says, okay, then I'm going to start the process of making cytotoxic T cells, all right, because that's how you fight viruses. Cytotoxic T cell is an interesting cell that can go right over to any other cell that has those proteins, and he doesn't need it to be displaying all of the other signals that I'm a sick cell early on in the viral respiratory process. He can actually make that cell commit apoptosis and get him gone. And so this is how you fight viral respiratory infections, because cytotoxic T cells, when they find a target, they send a signal back to the T helper cell and say, hey, I found another one, there's still virus here. We make more T helper 1 cells, more cytotoxic T cells, and the more of those we make, we destroy all of the nasty infected cells and they're gotten rid of. And when that happens, all the signals of inflammation go away, and then all of the macrophages and natural killer cells, cytotoxic T cells, die off. We regenerate all of those cells in our respiratory system, and in the last step, those T helper cells have become T helper memory cells. And here's where the magic begins, because that memory cell has been programmed by everything he was exposed to know that my job is to hang out in the respiratory system, and the next time I get stimulated with that protein again, I don't have to have Mr. Macrophage and Master Killer Cell and all that happen. I'm going to suddenly start making more cytotoxic T cells. We're going to go back to war, and that is why you have immunity, is that T helper cell. So a couple things you need to take away from this. First of all, cytokines are the big part of this problem. Regulating them is what drives this disease. Second of all, that cytotoxic T cells are how you fight a viral infection. Right? That's how you get rid of virus-infected cells. One of the things you have not heard me say once through this lecture is the word antibody. Now, this is very important to understand regarding antibodies. Can I have the next slide, please? So this is how you fight a viral infection, and you'll notice that there's not necessarily antibodies made. Antibodies just float in the bloodstream. They don't get out in between the cells. They don't have a way to tell you this cell is a bad cell and it needs taken care of. And your body doesn't like to make antibodies unless they're going to be useful, because it has to spend energy to do that, and there's risk to antibodies. But let's say you have a pathogen. Like another slide, please. Let's say you have a pathogen which is really good at getting out of the respiratory system. Let's go with mumps. Likes to get out very quickly, go to your salivary glands and your testicle, cause infection there. As those tissues start to become inflamed and they release their cytokines, some of those cytokines are actually sensed in the lung by the T helper cells, and they will turn to T helper 2 cells. And some of those T helper 2 cells, if I can have the next slide, will actually make fewer cytotoxic T cells. That's not good, right? We're not able to fight the infection as well, but we'll tolerate that so the T helper 2 cells can make B cells, which make antibodies, particularly the IgG, IgA class. And these things float through the bloodstream and take up all the little viruses that are circulating, so they can't take on any other tissue in the body. So this system is a worthy sacrifice when you have a pathogen that rapidly leaves the respiratory system and goes to other parts of the body. It's not worthy unless you're doing that, all right? But there are ways, for instance, if you were to take a vaccine that almost never got to the respiratory system but went through the entire body and suddenly made the entire body see that I'm getting infected in all my different tissues, where you could have a sudden expansion of many T helper 2 cells, a marked reduction in cytotoxic T cells. And this may lead to an enormous amount of production of antibodies, but antibodies don't fight viral infections. And now what you have is a situation where actually the virus can replicate in the respiratory system unchecked, generating more and more variants, some of which will be good, some of which will be bad, causing more in my cytokine production, which is going further and further out of control, and the antibodies don't have anything they can do to stop that. This is particularly a problem if you have what we call an enhancing antibody. So antibodies can be neutralizing, meaning they bind to the pathogen and make it less able to infect cells. But there are some variants to develop in this process that are what we call enhancing antibodies. Next slide, next click if you would. And enhancing antibodies actually can bind to the virus and make it better at infecting cells. And this isn't a theory. In the 1960s, we actually had a vaccine program that was designed against respiratory syncytial virus. And it's very interesting what we saw with that, which is what we see whenever we see enhancement developing. So it started off and things looked good for the people who were vaccinated. They actually had fewer cases of RSV than the people who were not. But then after about 69 months, there was a small increase that went up really quickly in the unvaccinated, and then a remarkable increase in the number of cases of the vaccinated. And when we looked at them and seen what had happened, we found out that there was a strain of RSV that had developed And it could develop because there was no war going on in the respiratory system that made an enhancing antibody that made RSV so it could actually infect cells better than it would before. And it ended up with more dead kids in the vaccinated group from RSV than in the placebo group. This isn't the only thing that can go wrong if you don't signal everything right to your immune system, because you can also get this thing, next if you would, where you can have it make an antibody called IgE, meaning the B cell has been told we're not fighting viruses, we're fighting a parasite. How many people here get runny nose, stuffiness, and sneezes when they're out there in the allergy season? Well, the reason you're having that is because your immune system has incorrectly been told that that piece of pollen is a parasite. Now understand what's happening is you have uncontrolled viral replication going on in your respiratory system, and you're developing an allergy to the virus as well, and the allergic reaction doesn't help you get rid of the virus. This, by the way, happened with 4 different types of vaccines directed against the SARS and MERS coronaviruses. This is what we call antibody-dependent enhancement. We've actually seen it twice in humans. We had another vaccine trial done with dengue virus where they didn't do all the proper research that developed the same problem. But there's even more things you can do wrong with this. Remember how important this is that the macrophage tells you what's going on from the infected cell? But if you don't give the right signals to Mr. Macrophage, or if you have a healthy cell displaying an abnormal protein who is giving no signals, then that T helper cell can get signaled under the right conditions to become something called an EXTH17 cell. This is actually an abnormal T helper cell who thinks he's fighting an extracellular bacteria and is very badly regulated, and he causes autoimmunity. And this isn't theoretical either. The last time our government tried to save us from a virus infection was the swine flu campaign in the 1970s. And after they started that vaccine, they actually started to notice it was causing the autoimmune disease Guillain-Barré syndrome at a greater rate than just the infection would itself. So, one of the things you should know is what we have done, because vaccines can go bad, all right? They can be very bad treatments. Is there has been a process designed to make sure that we didn't give out a vaccine which was going to cause autoimmunity and antibody-dependent enhancement. And that process was that you had to start off with animal trials and you had to carry them out for 2 to 3 years. They had to be placebo-controlled and randomized so that if we started to see antibody-dependent enhancement developing in the animals, then we could pull that vaccine back before it got into humans. By the way, this was, when we do these studies in animals, there was a vaccine program done for the viral disease in chickens called Marek's disease that was able to wipe out chickens very quickly. Showed the same phenomenon, by the way. The unvaccinated initially started to get worse than the vaccinated, and then it just changed hands, and the vaccinated chickens got smoked. Secondly, if you made it through 2 to 3 years of animal trials, then you could take small groups of humans and you give them increasing doses of the vaccine. We could study what all organs they went to. We could follow those human beings out in their small numbers for 2 to 3 years, see what happened to those organs. And if nothing bad happened then, you could do a large randomized placebo-controlled trial of human beings for a target population. What that means is if you were planning to give this vaccine only to people who didn't have blue eyes, then you had to exclude blue-eyed people from the test group. And then when the vaccine was approved for use in humans, you couldn't give it to anybody with blue eyes, all right? And then you had to carry those trials out for 2 to 3 years so we could make sure that it wouldn't develop antibody-dependent enhancement and autoimmune diseases in humans. And then after that, you would be allowed to expose it to the human population as a general treatment. And does anybody know how many of those safety steps were skipped in the making of COVID-19 vaccines? Every single one of them. That's right. So these are the basics, things you should take away from this. First of all, vaccines can hurt because vaccines have to, if you're going to do a viral thing, you have to make sure you have an infected cell, which is actually giving the right signals. You have to make sure you make cytotoxic T cells, not gobs of antibodies. We actually know when somebody is developing antibody enhancement, when you look at the population, they'll actually have higher antibody levels than the people who are unvaccinated. Antibodies don't mean immunity, they just mean you've been exposed. And you have to understand that if you don't present all of the information to the immune system right, it is very easy to confuse it and have it start fighting parasites or extracellular bacteria and causing off-target injuries. So these are the basics of viral infection you need to know so that you can go on and have understand the data that you're going to have presented to you the rest of the day. Any questions?